Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So we were discussing cell biology, in which we have started cellular signaling part, where in the last class we discussed about the GPCRs, G protein coupled receptors. So we have already discussed about the basic way the signaling works and what is cell signaling and what is the pathway that a cell has to follow so that a signal is received and it is relayed and the effector proteins are formed, right? So we started learning about receptor proteins so that this signaling pathway is clear to us all. So we learned about the G protein coupled receptors, which are cell surface receptor in last class. So we discussed that how the G protein, which is being coupled with receptor, if that G protein changes, the signaling also changes. So we listed down four different types of G proteins in last class. The stimulatory G protein, which is denoted as GS, the inhibitory G protein GI, the G12, G13 protein, and GQ. So these are four different types of G proteins, which, when being coupled with a 7-pass transmembrane receptor, result into different types of signaling. And that is why the names are also different. The stimulatory GS protein stimulates the adenylyl cyclase. Okay, so we learned about the signaling pathway in detail in last class, how the GS protein, it stimulates adenylyl cyclase. On the other hand, the antagonistic to GS protein, the GI protein, it inhibits adenylyl cyclase. Okay, let me just rewrite it. So GI, it inhibits adenylyl cyclase. So we already discussed this adenylyl cyclase. This enzyme converts ATP to CAMP, cyclic AMP, right? So as the by with the help of GI protein, the adenylyl cyclase is being inhibited, and that is why the levels of the CAMP, this secondary messenger cyclic AMP, will be reduced inside the cell, and that is why the Cre genes transcription will be inhibited, right? So this is the basic schematic of how the signaling pathway goes on. So what happens is that whenever extracellular signal comes, it binds to the receptor protein, the 7-pass transmembrane receptor. Then that if that protein is activated due to some conformational change due to the ligand interaction, so that conformational change results into the activation of GEF, the guanine nucleotide exchange factor. So now this GEF, it will replace the GDP, which is bound to the GI protein. So the GI protein, which was initially bound with the GDP, due to the Jeff, which is working on it, it will replace this with GTP. The GDP will be replaced with the GTP. And on the GTP, now the gap will work. The GTPase activating protein, which is the regulatory GTPase, okay, the regulatory protein. So this gap will work on this GI protein, where the GTP will be hydrolyzed, because this GI protein, which is a heterotrimeric protein, which has alpha, beta, and gamma subunits, the alpha subunit has the GTPase activity, right? So this G protein, it has intrinsic GTPase activity, but for activation of that GTPase activity, you need some external factor, the external protein, a regulatory protein, which is gap in this case. Okay, the GTPase activating protein is the regulatory protein. So this is going to activate the GTPase activity of alpha subunit of the GI protein, and that is why this GI protein will split into alpha subunit and beta gamma subunit. Okay, so this alpha subunit now, which has been activated due to the GTP hydrolysis, this is going to inhibit adenylyl cyclase. Okay, and due to the inhibition of adenylyl cyclase, now the CAMP levels will really decrease in the cell, and that will result into the muscle relaxation. So it has exactly opposite effect of what GS shows inside the cell, right? So we discussed GS in yesterday's class, and that is why I summarized the GI protein in today's class, right? So this is how the signaling pathway goes for the GI protein. So in last class, I also mentioned that not only G alpha is it is taking part in signaling, but in some cases G beta and G gamma, this G beta gamma subunit, they also have some downstream effect. So we did not discuss that in GS protein because there it did not have any significant signaling activities. But in case of GI beta gamma subunit, where the G alpha decreases the CAMP level by the inhibition of adenylyl cyclase, GI beta, they also have their downstream effects. What do? They open potassium channels in the cell, which results into the hyperpolarization of the nerve cell, right? So we discussed in neurobiology class how the cycles goes on, how the action potential is generated, right? How the depolarization of cell is done. Okay, so there we learned that depolarization, it is the reduction in the charge separation, and that is done due to the opening of sodium ion channels. But if we open the potassium ion channels, that is going to result into the hyperpolarization of cell. Okay, so we haven't covered the neurobiology series completely. So when we will learn about the vision sensation, at that point of time, I'll let you know about this signaling cascade because this is very, very important. Okay, so just remember, so those lectures, those cell biology lectures are very important. Here we are just, we are just getting introduced with the basic signaling pathways. Okay, but what downstream effects they will have inside the cell, we'll learn them in some other series like developmental biology, or let's say in neurobiology itself. Okay, so we'll learn about the downstream effects in those lectures specifically. So then I will mention that this is GI signaling or this is GS signaling. This receptor is a GPCR. So at that point of time, what I want to convey should be clear to you all. And that is why we are learning about the basic pathway, basic signaling cascade that goes on inside the cell, right? So today's point of focus will be the G-cube signaling. So I explained GS and GI signaling. We do not need to go in details of G12 and G13 signaling, but just for your information, it's just as a extra information. Know that G12 and G13 are those proteins. They are when they are activated, when those type of GPCRs are activated, they result into the cytoskeleton movements. Okay. Actually, the GIF which works on G12 and G13 is different. The Rho GIF, the Rho GIF works on this G12, G13 protein. We are not meant to learn this protein in detail. We will be learning about GQ signaling in detail today because the GQ signaling is very important because it is very crucial because this uses a phospholipase. Okay, and now I will mention a lot of points that we learned in the plasma membrane lectures when I was introducing you the plasma membrane, the structure of plasma membrane, the composition, the asymmetry that plasma membrane has, and how it is very important functionally. In that lecture, we discussed a few things that are going to be revised in this lecture now. So for that, let's first learn how the GQ protein it helps in the signaling, how the signaling goes on inside the cell. So, just like other GPCRs, it is also coupled this GQ protein when it is being coupled with a 7-pass transmembrane protein. If the signaling molecules arise and it activates the receptor, the GQ protein which was initially bound to GDP, the GDP will be replaced with the GTP, and now this GTP will be hydrolyzed with the help of GAP. The GTP is activating protein, and once this GQ is hydrolyzed, just like any other G protein, it will be also hydrolyzed into alpha subunit and a beta gamma subunit, right? So this alpha subunit is crucial for downstream signaling because it is going to activate an enzyme known as phospholipase C beta. Okay, and this is very important because okay, so from the name of the enzyme, it is quite evident that it is a lipase, means it is going to break down a lipid molecule. So it is going to break down phospholipid because it is a phospholipase, right? So the phospholipid that it breaks down is the phosphorylated phosphatidyl inositol, and I have mentioned this in previous lectures as well. Okay, the phospholipase C beta, the working of phospholipase C beta is very important in some downstream signaling pathways. So it breaks down a molecule known as PIP2. Okay, so PIP2 is phosphatidyl inositol 4,5 bisphosphate. Okay, so this is actually a phosphorylated phosphatidylinositol. So phosphatidylinositol, when it is when the two phosphate groups are being added on phosphatidylinositol, it results into formation of PIP2, the phosphatidylinositol 4,5 bisphosphate, and this is broken down by phospholipase C beta. Okay, so this is very important. So that you remember when we were discussing about functional asymmetry of the asymmetry of plasma membrane, we discussed that how it is important for the functioning of the plasma membrane. Why this asymmetry is important? In that lecture, we discussed that the presence of phosphatidylinositol, the unequal distribution of phosphatidylinositol in the cytosolic as well as exoplasmic leaflet of the plasma membrane is very crucial for the signaling. So now you remember why it is crucial, because this phosphatidylinositol, which is present unequally in the outer and inner monolayer, it is very important because it is going to do the intracellular signaling now. Because the breaking the splitting of this PIP2, the hydrolysis, the breaking of this PIP2 by the phospholipase C beta, this reaction is going to trigger some cytosolic signaling, and that is why this is how the asymmetry of plasma membrane plays a very important role, right? I hope you remember all of that part that we discussed there. So this PIP2, when the phospholipase C beta works on this PIP2, it is broken down in two parts. Okay, the IP3, which is the inositol 1,4,5 triphosphate. Okay, it breaks down the PIP2 in IP3 and DAG. So I hope now you remember I discussed that diacylglycerol, DAG. Diacylglycerol is a secondary messenger, right? So it is also going to do some downstream activities. Okay, well, we'll come to this later. Let's first focus on IP3. So this both of them, IP3 and DAG, both of them are going to do some downstream signaling, but the difference is DAG, diacylglycerol, is lipid soluble. I mentioned this in last class as well, the signaling classes, and whereas the IP3, it is water soluble. Okay, so as IP3 is water soluble, once the PIP2, imagine this, close your eyes and imagine what I'm trying to convey to you. PIP2, which is sitting inside the inside the plasma membrane, on the cytosolic leaflet maybe, the phospholipase C beta comes and breaks it down. Okay, so imagine phospholipase C as a scissor. It is cleaving, it is severing the PIP2. So it is chopping off PIP2. When it breaks the PIP2, the lipid soluble part stays in the plasma membrane. It is the diacylglycerol. Okay, so diacylglycerol being lipid soluble, it stays in plasma membrane, and the other part, which is water soluble, which is being chopped off after PIP2, it just gets released inside the cytoplasm. Why? Because it is water soluble, so it doesn't stay in plasma membrane. It is the hydrophilic part of the PIP2 that gets into the cytoplasm because it is water soluble. So this gets, when it gets into the cytoplasm, it is going to do the intracellular signaling activities because it is now has been introduced in the cytoplasm. So what does it do? That we are going to now learn in detail. So IP3, it now will act as a secondary messenger. It will act as a signal molecule or a ligand for some receptors that are present on the membrane of the ER. Okay, so now just imagine this whole scenario. PIP2 is being cleaved by phospholipase C beta. The TAG stays in the plasma membrane. We'll come to DAG, the diacyl glycerol later. So now this IP3 is being released into the cytoplasm. It will wander in the cytoplasm and now it will go and bind to the receptors that are present on the endoplasmic reticulum. Okay, so the receptors that are present in the endoplasmic reticulum for IP3, they are obviously called as IP3 receptors. Okay, so those IP3 receptors are actually a large IP3 gated calcium channels. So these are calcium channels. Okay, the calcium channels which has affinity for IP3. Okay, the receptors for IP3 means the receptors are what? The proteins which are going to interact with certain molecule. So if they are let's say X receptors, means these are the proteins which are going to interact with X ligand, right? So these are IP3 receptors, but they also have other characteristics that they are calcium ion channel. For example, you are a student, but you also, you are a 10th standard student, but you also teach fifth or sixth standard students. So you are a student, you are a 10th standard student, but you also have other role. You are also playing a role of being a teacher, tuition teacher for fifth and sixth standard students, right? So you have two roles, two different roles. Okay, so in the same way, IP3 receptors, which are the receptors for IP3 molecule, they are also calcium ion channels, and that is why we call them IP3 gated calcium ion channels, because they will only open when the IP3 goes and binds, goes and interacts with them. Okay, so this proteins, but here there's a very interesting fact that not only the IP3 will trigger the opening of the calcium ion channels, but for the opening of the calcium ion channels, this IP3 gated calcium ion channels, you need to fulfill two requirements. First is IP3 binding. Okay, so now this IP3 binding to those channels will trigger some conformational changes. So now this conformational changes will do what? It will expose the calcium binding site of this receptor proteins to the, towards the cytosolic part. Okay, let me just draw it schematically so that it will be clear to you all. So this is a plasma membrane, right? And this is the nucleus, right? And this is ER. Okay, okay, so this is the ER membrane. This is our nucleus. Okay, this is nucleus. This is cytoplasm. This is the plasma membrane, and this is the membrane of ER. So on the membrane of ER, the IP3 receptors are situated. Okay, so that what happens is that this membrane of ER has again. So let me just zoom into this. So membrane of this ER has a cytosolic side and a side which is facing the lumen of ER. So this is the lumen facing side. Okay, so as you, imagine this as a 3D diagram. So this is cytosolic side, and this is the side which is facing lumen of the ER. So this is the ER. So this is lumen of ER, and this is the cytosolic side, right? So what happens is that when the IP3, so here, okay, so here this was PIP2, it gets chopped off, right? So DAG remains, okay, sorry, let me just change the color. So DAG remains on the on the plasma membrane, and IP3 gets into the cytoplasm. So this IP3 will go and it will bind to this receptor which are present on the endoplasmic reticulum membrane. So now what will happen, this IP3, as I already mentioned, those are calcium ion channels, right? So this means that they will open and they are going to do the exchange of the calcium ions. So they have, those ion channels have a property of having a binding site for calcium, but it is not exposed towards the cytosolic site. It is actually hidden inside the protein. So due to when the 3D structure of protein, the 3D structure of those receptors is somewhat like they are hiding the calcium ion binding site inside them. Binding of IP3, when IP3 comes and binds to those receptor, binding of IP3 triggers some conformational change, and that conformational change will result into the exposure of the calcium binding site towards the cytosolic part. Let's say that this was a protein. Okay, this was protein, and inside this was the calcium binding site, which was well covered, well hidden inside the protein. But due to binding of IP3, so now IP3 came and bound here. Okay, so due to binding of IP3, some kind of conformational change will happen, and let's say that this protein now changes like this, something like this. Okay, I know I'm not drawing it correctly, but this is just for your understanding. So it is exposing calcium binding site, right? Okay, and IP3 is already bound here. So this is IP3, and this violet one is the calcium binding site. Okay. So even though the calcium ion concentration inside the cell it is very low, it is I don't remember the figures, the digits correctly exactly, but it is 10 raised to minus 7 micromolar or something of that sort. Okay, so the what I want to say is that the concentration of calcium ion inside the cell, it is less. But even though it is less, the calcium binding site which is present in those IP3 gated calcium channels, it is that binding site has higher affinity for calcium. The concentration is low, but it is sufficient. It is enough for some calcium to go and bind here. Okay, even though concentration is less, but we have sufficient amount of calcium present inside the cytosol, so that that calcium will go and bind to this calcium binding site. So we need two conditions for the IP3 gated calcium ion channels to open. First is that IP3 should go bind to that receptor, and it should trigger the conformational change. And once it triggers the conformational change, the calcium binding site should be exposed on the cytosolic part, and the cytosolic calcium will go bind to those calcium binding site present on the protein. And now when both IP3 and calcium, they are bound to this, so now when both IP3 and let's say here calcium bound to this channel, to this protein, when both of them are bound, both of those conditions are fulfilled, only then this IP3 receptor ion channels, calcium ion channels will open. Okay, so once those calcium ion channels, IP3 gated calcium ion channels or endoplasmic reticulum, they open. Okay, when they open, what will happen? They will do the calcium ion exchange. So calcium ion influx will be seen. Okay, influx means here, whenever I'm saying influx here, this means that the calcium ion is being released from the endoplasmic reticulum inside the cytoplasm. Okay, so it is being released inside the cytoplasm, and that is why we are referring influx. Whenever a certain molecule is being taken inside the cell, it is influx. Okay, though in the case here it is being thrown out from the lumen inside the cytosol, so it is being released inside the cell. So whatever result will be seen, that the calcium levels inside the cell in the cytoplasm. Okay, so inside the cell, the calcium levels will increase. Okay, so as the calcium level increase, what will happen? This calcium will go and bind to a calmodulin protein. So the four calcium ions goes and bind to this calmodulin protein, and which will, so this protein is also known as calcium binding protein, the calmodulin. So when the calcium goes and binds to it, it will activate an enzyme known as CAM kinase, which is going to do the Cre gene transcription. Okay, so this is not a point of focus right now. So whatever downstream effects calcium is going to have, we will discuss this again in some other series, right? But what I wanted to, what is point of focus of today's lecture is how the GPCR signaling goes on. So whenever I mention GQ signaling or calcium, whenever I say with there's a GPCR situated on some cell, and that is going to result into increase in calcium ion level. You should immediately know that that was GQ signaling, right? The phospholipase C beta is going to act, and that is going to result into the increase of calcium ion levels inside the cytoplasm. So once those calcium ion levels increase inside the cytoplasm, that is going to result into muscle contraction, right? So this was the IP3 part. Now let's come to the diacylglycerol part. So what happens is that due to the signaling of IP3, okay, the calcium ion increased, right? So this increased calcium ion, okay, this increased calcium ion levels, the presence of DAG, as well as the presence of phosphatidylserine. I hope you remember that phosphatidylserine also has unequal distribution in both leaflets, in the cytosolic and the exoplasmic leaflet of the plasma membrane. So the phosphatidylserine, okay, which is present on the cytosolic part of the leaflet, okay, on the cytosolic part of the plasma membrane, those three together, they are going to show some downstream signaling. Okay, so what, let me just summarize it again. The DAG, which is present on the plasma membrane, plus phosphatidylserine, again, which again has unequal distribution on the plasma membrane, and the calcium ions, which are, which are now have high level inside the cytosol due to IP3 signaling. All of them, all those three are going to show some downstream effects, which again are not the point of focus of this discussion, but I will point this out in some other lectures in the future series. That point of time you should remember, right? So DAG also has some downstream effects, as well as IP3 also has the downstream effect, and that is the increase in level of calcium ions. Okay, so let's now just summarize the whole discussion. Okay, I have a schematic from the Bruce Alberts again. So this simple schematic is summarizing all of the lecture, that all of the things that we discussed today. So let's summarize it once. So this is a 7-pass transmembrane protein, the receptor, the GPCR, the receptor which is being coupled with GQ protein. Okay, so this receptor, when it receives some extracellular signal molecule, right? I wanted to mention one fact, that when we will discuss about, there's some other series development in developmental biology or even in cancer biology, we will be focusing on those signal molecules as well as those downstream effects that those signaling pathways have, right? But for the purpose that you should know how the signaling works, those series I've started this series where we are discussing about the basic receptor signaling, right? Okay, yeah. Well, the signal molecule will come. It will bind to the GPCR, the receptor protein, and now it will trigger some conformational changes in the receptor protein, right? So those changes and conformational changes will result into the activation of GEF, the guanine nucleotide exchange factor, which is going to replace the GDP, which was bound to the G protein. So now in this case, it is GQ protein. So GQ protein, to this GQ protein, the GDP was bound, and that will be replaced with the GTP, right? And due to the replacement of this GTP, so now when the GTP hydrolysis is done, the alpha GTP will, the subunit will be, so the G protein will split into alpha subunit and a beta and gamma subunit, right? So this alpha subunit, which is GTP bound, once the GTP is hydrolyzed, it is being activated and it is going to do some downstream signaling. So what does it do? It goes and activates the phospholipase C beta, right? Once this enzyme phospholipase C beta, so it is evident from the name that it is an enzyme which is going to hydrolyze a phospholipid. The phospholipid it works on is the PIP2. Okay, phosphatidyl inositol 4,5 bisphosphate, right? So this is PIP2, which is situated on the plasma membrane. So here you can see it has some hydrophobic part and it has some hydrophilic part. So this PIP2 will be hydrolyzed by phospholipase C beta into two parts: the diacylglycerol, which is lipid soluble, which will stay in the plasma membrane, and the IP3, the inositol 1,4,5 triphosphate. So this IP3, being water soluble, will be released inside the cell cytoplasm, and it is going to go and bind to the IP3 gated calcium ion channel receptors. Okay, so those calcium ion channels, they are IP3 gated. So IP3 will go and bind to this as well as calcium. So the calcium, so that the conformational changes will be seen. The calcium binding site will be really, it will be exposed towards the cytosolic part. So now calcium is also bound to this channel and IP3 is also bound to this channel, and due to the binding of those two molecules, IP3 and calcium, so now this IP3 gated calcium ion channel will open and it is going to release some calcium ions inside the cytosol from the lumen of the endoplasmic reticulum, right? So whatever calcium is being released, it is going to result into a lot of things such as muscle contraction, right? But the calcium which is released, so now overall concentration of calcium inside the cytoplasm is increased, right? So whatever calcium is being released, out of that, some calcium is going to activate protein. Yeah, well, I did not discuss this. Let me discuss this. So it is going to activate a protein kinase C. Okay, yeah. So the protein, what is, so no, not just, but for the activation of protein kinase, I told you that DAG, which is present here, right? DAG, presence of calcium, so high amount of calcium, as well as the phosphatidylserine. So three things: DAG, phosphatidylserine and calcium. Those three things, they are going to show some downstream effects. So what downstream effects they show? They activate an enzyme known as protein kinase C. Okay, so this enzyme is named as protein kinase C because it is calcium dependent kinase. So this kinase so this is a kinase, so this is going to phosphorylate certain protein and it is going to result into some kinase cascade. Okay, so this is also acting as a, so this is also will act as a molecular switch. So we'll discuss about this in future. But for the activation of protein kinase C, we need three things: the DAG, the calcium ions, as well as the phosphatidylserine, which is present on the cytosolic part of the plasma membrane, right? So no, if one of those things are not there in the plasma membrane or not in the cell, the activation of protein kinase C will not be seen. So for the protein kinase C to do its job, to do this downstream signaling, we need the calcium ion activation of the protein kinase C, as well as we need the DAG to which it can bind, as well as the phosphatidylserine. Well, the phosphatidylserine is not shown in the schematic, but it is also important because we have already discussed this about this in the plasma membrane series, how the functional asymmetry is important for the functioning of a cell, right? So you can refer to that lecture if you have no idea about the asymmetry of plasma membrane. So with this, I conclude the lecture. We have discussed about the GPCRs, the G protein coupled receptors in detail. So we are done with GPCRs discussions. In next lecture, we'll discuss about receptor tyrosine kinases, the enzyme coupled receptors. So as I told you that there are three type of receptors: the cell surface receptors, and there are some intracellular receptor, right? The three main categories of receptors are there. Some are enzyme coupled, some are ion channel coupled, while some are G protein coupled. So we already discussed about G protein coupled receptors. We will discuss about enzyme coupled receptor, and we have already discussed about ionotropic receptors or ion channel coupled receptors in the neurobiology series. Okay, so I conclude the discussion here. In next class, we'll discuss about RTKs. Till then, see you. Bye bye.